So moving on now to, I believe this is a joint talk between Laure and Michela from OpenAnalytics and Galapagos. Take it away.
Yeah, indeed. So thank you for the nice introduction. Can you see my screen up and the presentation correctly? Yes, I guess so. Yes, thank you. Okay, thank you very much for the organizer of the Pharma conference to allow us to present a use case, how we use R to create interactive medical oversight reports in R, indeed. So this is a joint talk by me, Michela, and Arne. I will first start with a short introduction to the medical monitoring, then I will demonstrate the tool on the publicly available datasets, and then Michela will show the overview of the different functionality of the medical monitoring tool. So this project is really a collaboration between Galapagos and OpenAnalytics, which are respectively a biotech and a data science company from Belgium, and all the slides are available here if you are interested to have a look. So what is medical monitoring first? So medical monitoring or oversight consists to explore and review safety data collected during clinical trial. So this is typically a task which is done by the medical monitors. And the goal is really to detect as early as possible safety signals. So the goal of the project for us was to develop an interactive in-house tool with a standard set of visualization and table of raw safety data, which should handle full customization. We should be able to handle both blinded and unblinded data sets. And it should be, of course, user-friendly enough for the medical monitors. So there is an example monitoring report available here. Feel free to have a look after the presentation if you would like. This is based on the original pilot study dataset from CDISC. So we use as input SDTM dataset available in the GitHub of the FUSE project. And the goal of the study was to investigate the safety and the efficacy of a drug, xanomeline, in patients with mild to moderate Alzheimer's disease. And in terms of safety, they were mainly interested about adverse events, vital sign, and laboratory evaluation. So I will go quickly to the report to show what it looks like. So this is a HTML document which contains multiple sections. So one section of interest for the medical monitor, especially the number of subjects which were enrolled in the study. And in this representation, you can see the number of subjects enrolled by country and for the different sites. So you could see there are 254 patients which were enrolled and dispatched in multiple sites. Another overview is a demographic table, which is typically used for clinical trials. So you can see the patients were actually randomized to 3 different treatment groups, so placebo, xanomeline, high dose, low dose, etc. I will go now to a different section, which is also very interesting, I think, for the medic. It's the adverse event section. So in this report, we display in an interactive table a summary view of all the adverse events which occur during clinical trials. So all the tables are interactive, but there is also the possibility to export them to a more static format, which is more like CSR compliance that you can see here in the Word document, which is opening. Okay. And then for each table and visualization in the report, there is also the possibility to dig down to a specific subject of interest. So in this case, where you can see here one subject which had an adverse event which resulted in death. And in this table, you can have a look at the specific patient ID. And for each patient, you can display a specific report that we also created, which is called the patient profile. And the idea of this patient profile is really to visualize and to give a global picture of what happened for a subject during the study. So in this case, for the subject, it displays some demographic information here, some medical history information, and most importantly, which treatment the patient got during the study, and how can this be linked, for example, to the adverse event or concomitant medication. So you can see, for example, this patient got treated twice with xanomeline and finished his trial at 60 days. And at the end, there was a sudden death. And you can also see how this can be linked to some laboratory test measurement in the reports. Another visualization which could be of interest is to have a general overview actually of all the adverse events which occur during a study. So for that, we display it in a tree map visualization, which looks like that, which represents the number of events per adverse event term subject organ class. So you can hover on the plot, and you can also select a specific class of interest. And if you click on P, like patient profile, you will be able actually to download all the patient profile for this specific set of interests. Another functionality that we have in the report is the possibility to compare multiple data batches, because typically in a clinical trials, we receive different interim data sets all along the clinical trials, and we're interested to see which records are new, which records have changed, and it's what we try to represent in this interactive table. Maybe to finish, I will just quickly show different visualization that we have. So it's time profile of specific laboratory parameters. So it's currently loading. So for each visualization, in the report, we also have an attached table linked to it, such as actually the medical monitor can, for example, select a specific patient. So let's have a look at the same patient I was showing previously here. And only the record for the specific patient are indicated here. So I can select all these records in the table, and then they will be highlighted in the visualization attached to it. Okay, so I will let the word to Michela to display you all the different functionality.
Thank you. And the second part of the presentation then aims at giving you an overview on how the tool you've just seen is produced. So let's go. We start with the minimal data processing. So we receive STTM data from the CRO, and we do a minimal derivation that we call STTM plus, in the sense that we just derive the analysis set, and we do some variable formatting. Then the data go into the package, into our custom package, and we produce the HTML. So the package contains in itself some templates, and each template creates sets of chapters. There's also the possibility to provide for the user a custom template anyway, but the key of the package, we think, is the use of configuration files that are in a YAML format. So configuration files allow the user to not really dig into R, but just to set up as a shopping list the parameters specific to the study at hand. So then it results that each configuration file corresponds to a chapter in the HTML, and bunches of configuration files are handled by the same template if they belong to the same group. So to give you an example, this is a configuration file that can be set up via, for instance, calling the default template in our package, and then the user has just to set the title, the data to retrieve, potentially some filtering of the data is required, and then to set up the list, to write the parameters that are needed for the table. And the mirror of the YAML file is the JSON schema, because we handle the validation of the input parameters and the documentation via JSON schema that I can't deepen in this call, in this presentation, but if you don't know it, it's a very interesting tool. And in a way to wrap up, we've seen several functionalities like interactive visualization, patient profile, tables for descriptive statistics, comparison of data batches for an ongoing trial, and there is also the possibility to add a specific alert for the study. And all this is created via custom functions around Plotly, DT, FlexTable, CompareDF, and through custom packages developed in-house as patient profile visualization and in-text summary table for the CSR-ready descriptive statistics. I would like to also mention how we care about quality control. We are working together with the Q&A department for spinning up a qualified R environment, and at the moment we have a unit test, and we assess the coverage of the unit test with the COP-R package. We use Git for code versioning and Jenkins as a CI/CD platform for the automated builds and checks of the package. And all these tools, fully customizable, that we just shown you, we are developing to reply to the needs of medical monitors and for early QC of the data. And if you are interested, there is the link available for the demo. And if you are interested, please reach out to us. We would be very happy to reply to any question or feedback either via email or in the chat. And thank you for listening. Thank you for the organizer of the conference.
Okay, yeah, great. Thanks, Laure. Yeah, so we have plenty of time for additional questions. Arne has been doing a great job answering a lot of those in chat. So I guess.
Welcome.